All right, so today we're going to talk about meiosis. So meiosis is one of the three types of cell division. So if you remember, mitosis is when we need to make more cells so the cells divide. Why do we need to make more cells? Well, humans are growing. They need to repair cells, that kind of stuff. But there's a second type of cell division called meiosis. And meiosis is a specific type of cell division. And so we're going to talk about that. So the main question is, why do eukaryotes need a second way to divide cells? What is the reason for meiosis? So the next couple slides we should be able to answer that question. All right, so before we start, we should know the difference between somatic cells and gametes. So somatic cells are all your body cells except for your sex cells. So the two sex cells are egg and sperm cells. So egg cells in females and sperm cells in males. And so all these undergo mitosis. So everything like your hair, your nails, your skin, your organs, all those, except for your sex cells, undergo mitosis. Gametes, though, undergo meiosis. They undergo a different type of cell division called meiosis. So here in this picture, you actually see a sperm cell fertilizing an egg. All right. So all somatic cells are diploid. What does diploid mean? It means they contain two sets of chromosomes, one from mom and one from dad. So we have 46 chromosomes, 23 from mom and 23 from dad. So diploid di means two. Haploid cells, that's when you get half. So all gamete cells are haploid. So in humans, the haploid number is 23. And so if you think about it, you only want 23. Why? Because mom will give 23 in the egg and dad will give 23. If they both gave 46, then that offspring would have way too many chromosomes. So diploid versus haploid cells in your body have a complete set, all 46. Gamete sperm have 23. It's called haploid. So what is meiosis in general? It's a cell division of the gametes. Remember, you have half a number of chromosomes. It occurs in sexually reproducing organisms and ends with 4 gametes containing 23 chromosomes. It can produce many types of offsprings. So why do eukaryotes need a second way to divide cells? Well, because the sex cells, the egg and the sperm cells, they only want half, which is called haploid. And so mitosis, you double your chromosomes and then you divide. And so you always get the same number of chromosomes, which is important when you want to make new cells. But if you want to create offspring, you only need to give half your DNA. So sperm and egg cells, they only have half the DNA. Okay, so the next question we're going to ask ourselves is, what are the steps of meiosis? Why do siblings look different? So what is meiosis? It's similar to mitosis, but will produce 4 non-identical daughter cells. So in mitosis, this is really good here. What happens is, if you were in interphase, so this is 2 N reverse diploid. So diploid means you have all 46 chromosomes. And then it actually doubles, remember, in the S phase, in interphase, it doubles, and then it splits into 2 cells, each with 2N, or diploid, and that's what you want. But in this case, it doubles in the interphase, just like normal, and then you have to go through twice. So we call this meiosis 1 and meiosis 2. And then what happens is these 4, in this case sperm cells, are nothing alike. They all have different combinations of DNA. And we'll tell you why it's a little different. All right. Oogenesis, and we'll talk about later, spermatogenesis, is basically, Genesis means the creation of, right? And so oogenesis is how eggs are made, right? And what's interesting is that you make 4 cells, but actually 3 of them are not viable. So they can kind of just die off. They're called polar bodies. And so you make only 1 egg. So from one, they call it germ cell, you only make 1 egg. So this is the germ cell. However, for males, spermatogenesis, you make 4 equal sperm. They all are viable. Okay. So spermatogenesis, oogenesis are the names of the way eggs and sperm are made. So if you remember, meiosis is actually very close to mitosis. So if you remember PMAT, prophase, metaphase, anaphase, and telophase. And so what's different is in meiosis you actually have 2 steps, kind of like what we drew. And so, or this image here. Right. So here, remember you have meiosis 1, and here we have meiosis 2. Okay. All right. So we're going to go through meiosis 1 first. So meiosis 1, prophase 1, this is where the homologous pairs form and crossing over occurs. But let's talk about what a homologous pair is, and then we can understand what crossing over. So remember, this is your 46 pairs of chromosomes, and so remember each one has a pair, and so each pair, like this is what we call chromosome number 1. These are called homologous chromosomes. So the prefix homo means the same. So basically, like, one's from mom and one's from dad. And so we call them homologous because they actually code for the same genes. So you'll learn later in genetics, like let's say mom gave you the gene for blue eyes, dad gave you the gene for brown eyes. You're going to have brown eyes because brown eyes is a stronger gene we call dominant over the recessive or the less dominant blue eyes gene. So, but what I'm saying is the gene for eye color is all on that same. So mom gives you a version, dad gives you a version, but since they code for the same, or they're the same chromosome, that we call them homologous chromosomes. They come in matching sets, so to speak. So what happens in prophase 1? So still in prophase 1 is these homologous chromosomes, they line up. But when they line up really close together, they actually, you don't have to worry about this word, chiasma, but what they do is they cross over. So like part of, say, if green was dad and mom was red, like they actually switch spots. So now you have chromosomes that have, you can see has part mom and you got part dad, right? And so this is kind of really strange, but actually what it does is it increases the variability, because now you don't have a chromosome that's only mom. So like you don't like if you get chromosome 1 from mom, you only have mom's genes. You actually have a combination of mom and dad. So this is really important in terms of crossing over. All right, so what happens is homologous pairs line up. First of all, in prophase one, they they start forming and they pair up. Then the pairs line up at the equator, right? Meta means middle, and then anaphase they separate. Telophase they reform to form two new cells. But what happens is he goes through another round. Same thing though. Prophase 2, nuclear envelope forms, the DNA condenses, and then the metaphase, remember metaphase, they line up, anaphase, they separate. But now you're like having it one more time. And so what happens is you end up with 4 different cells. Okay. This is a really good review. So what are the steps of meiosis? Remember, you have basically meiosis 1, meiosis 2, and you have PMAT 1 and 2. Why do siblings look different? Well, what happens is, for 2 reasons. One, crossing over, but another thing called independent assortment. And what that is basically you randomly get, it's not like you get all moms or all dads DNA. Sometimes you'll get half a mom, half a dad, so it's all random. So there's random chance of you, it's a 50-50 chance that you get mom, you get dad, and then also you have crossing over. So the 2 reasons why siblings look different: 1, crossing over, and 2 is you get, we call it independent assortment. So like they sort out independently, so you get, it's basically a 50-50 chance, a random chance of receiving mom or dad's genes. Okay. All right, you guys can watch this if you'd like.